Age-related macular degeneration is one of the leading causes of blindness in the elderly population, affecting over 2 million people in the country.
For the atrophic or dry form, which represents about 90% of patients, there really is no FDA-approved treatment. And so that's really characterized by this progressive degeneration or atrophy of the retina pigment epithelium. So a very important cell type in the eye.
Patients essentially develop blind spots directly in the center of their vision. This makes it very difficult to read, even with magnification. Driving becomes almost impossible in the late stages of the disease. Patients may have difficulty recognizing faces. They may see someone but not be able to see the detail of their faces. Patients start to become depressed and isolated because of this.
Which raises the question of how do we develop better therapies and treatments in the field for our patients to help treat, prevent, and reverse blindness. It had been previously thought that age-related macular degeneration was primarily a disease of the photoreceptors that we see light with. However, our research showed that it's actually the support cells of the retina, the glial cells, which we call glue, and the vascular cells, which were causing the damage. And this changed the paradigm of how to develop new treatments for the disease because it allowed us to pinpoint the exact cell types in which we should target with new therapeutics.
In particular, using embryonic stem cells, as well as a technology called induced pluripotent stem cells. And so what that technology was able to do was essentially just use stem cells and revert them back into this embryonic stem cell-like state, and they're able to become essentially any cell type in the body. For us, we actually use those to turn them into retinal pigment epitheliums, the cell type that's mainly affected in macular degeneration. And we can essentially try to use those to replace the damaged cells. And then the second part of our laboratory is kind of a drug development arm. So we can use stem cell technology as well to be able to mimic essentially what we see in the pathology of the disease. And we can identify potential drugs and other therapeutics that essentially can help to reverse the disease.
So I think what keeps me in the work is the patients at Yale Medicine, to see people each week and to hear their stories of how macular degeneration affects them and affects their loved ones. My grandmother has macular degeneration, just to kind of see the effect on her life and how rewarding it could be to be able to have a new therapy that can really help.
I think vision is our most important sense. And I've been studying this for a very long time, and to see that we're coming so close to treatments and therapeutics that will have benefit for these individuals really excites me.
There is no better sense of fulfillment in this world, I think, than actually being able to make a qualitative difference in somebody else's life. There are a variety of strategies available for the treatment of macular degeneration that were not present 10 years ago. And there's no doubt in my mind that 10 years from now we'll be saying the same thing, or even 5 years from now we'll be saying the same thing.